of experimental design to consider is the depth or comprehensiveness of the microbial data to capture. This will depend on the question you're attempting to address and the diversity and the range of the richness of the microbial populations in your specimen. Sequencing-based methods require that you go surprisingly deep to capture a complete view of the microbiome. For instance, 10,000 to 100,000 reads typically won't capture moderate to low biomass numbers in a bacterial community. The figure shown in this slide from a recent study by Turnbaugh et al. Demonstrates that with about 100,000 quality reads, these were high-quality reads, they were able to observe only about 60% of the total phylotypes of a fecal microbiome. A hybridization-based approach has the advantage of capturing a much broader range of the richness of the microbiome because of the dynamics of hybridization kinetics inherent in the technology. For instance, in a study focused on new drug treatment discoveries, or one aimed at understanding response to treatment, you would want to obtain as much information about the microbial population as possible. There may be low or moderate biomass numbers triggering a cascade within the population that you would want to capture in your analysis. And the dynamics of hybridization are such that all of the members are competing equally for the probes in the assay. And so it's not sampling that you're doing. It's detecting everything that has a partner in the assay, that has a complementary DNA probe. And therein lies the sensitivity. So sample numbers. The purpose really of having enough samples is to manage sources of variability. You want to use your pilot and follow-on studies to characterize sampling bias and variation introduced by sample handling. You really want to understand this early in your path. You want to understand sample heterogeneity issues that could confound your biomarker panel and that you need to address. You certainly want to have a good understanding of specimen collection, storage, and shipping methods and how they may impact sources of variability. Delays in specimen processing can also be a challenge, but there are other aspects of specimen processing that must be balanced against that. So, for instance, managing batches and batch effects. You want to work with a laboratory that understands how that can impact data interpretation. And so banking samples, understanding that they're stable while they're stored, and then having them run so that, for instance, if you're comparing two groups, Group A and Group B, one of the things that you want to avoid is collecting data from Group A on Monday and collecting data from Group B on Tuesday. People recognize that this can introduce a batch effect that can actually confound your interpretation of what's due to handling of the samples versus what's due to biological variability. And then in the samples themselves, you want to make sure that you have well-annotated samples. That lab metadata can be incredibly important. Environmental factors such as diet, medication, habits, other factors, age, gender, reproductive status can impact your interpretation. So managing those and understanding those is important. It's important to realize that similarities and differences will occur due to chance. And when you're looking at a lot of information, you just simply see more of those things. A large enough sample number is needed to minimize or smooth the inherent biological diversity that's not the subject of the study and reveal true group characteristics. So in summary, biological replicates, in initial pilot studies, you should incorporate a minimum of 5 biological replicates per class, cases versus controls, time points, body locations, and the controls that could be informative for data interpretation so that you can extract the information at the end of the study that you care about. Technical replicates. If you're running your samples in a laboratory that's running good laboratory practice and quality management systems on a well-characterized platform, Technical replicates are not generally necessary. That's actually part of the lab's business. That's what the lab should be taking care of. They're running those types of controls, positive and negative controls, and they know about the variability. They should be managing batch control for you. So technical replicates are generally not necessary when the analysis is conducted in a laboratory that is set up to do these types of studies. Results of pilot studies inform power calculations of the next study. And keep in mind that this is an iterative process. So it's important to think in terms of sample accrual and having extra samples that you can add to your study if it looks like you need to add those to help really establish that biomarker panel that you want to take forward. So thinking in terms of a contingency plan that includes banking extra samples, and thinking in terms of the statistical power demonstration is an iterative process during the pilot study, and you'll want to continue to revisit that and test models where additional samples will challenge the signature. And when you get to the point where they don't change the signature, you've begun to arrive at establishing a biomarker panel. Remember, during the pilot, you are simultaneously discovering and testing the signature. So this takes us back to the overview and that where we are in the past. So you've conducted your pilot study, and in the discovery phase of your project, it's important to challenge the robustness of that putative biomarker panel. There are a number of methods for testing the signature, and a detailed explanation is beyond the scope of today's talk. But generally, you test the signature in a confirmation phase, where you're challenging the components of the signature and challenging the number of specimens used to reveal the signature. In one analysis method, you add additional specimens to determine if your putative signature changes. Does it plateau? Achieving a plateau suggests signature stability. Similarly, moving taxa in and out of the signature will aid in evaluating which taxa are critical to driving the differentiation between patient groups. There's many approaches for carrying out these analyses, and these include rarefaction, curve analysis, and cross-validation methods, such as leave-one-out analysis. Although some may consider these to be computationally expensive, my bias is it's better to invest early in these sorts of cross-validation studies to challenge the robustness, to later on in the pipeline or path to learn that your signature is not going to stand up. So don't hesitate, jump in. You've got a question that you really want to address. You've thought of something that would make medical treatment better or that would improve patient care in the clinic. So now's the time to go for it. But practice good scientific methods. We're happy to talk to you about study design and data analysis. They're very important to think about early and set up your plan so that you will be successful in identifying a robust signature. It's important to do this in alignment with clinical and laboratory best practices. You want to start thinking about what the expectations are in the clinical laboratory for the performance of the assay. You'll get to that. You'll get to establishing analytical performance. But if you align early, you'll accelerate your path. You'll accelerate your, you'll shorten the distance between discovery and introduction of the assay into a clinical, utility and validation. And then finally, and certainly not the least, is ensure that you have a robust analysis and data management plan in place, that you're working with people who are experienced in this area, and that you've thought through the volume of data that you're going to be collecting and the ways that you'll be annotating the data so that you can extract the most meaningful information possible. So, Rachel, that's all I have, and I'd like to pass it back to you.